Hi, I'm Neil Greenspan. I'm a professor of pathology at Case Western Reserve University and senior editor at Pathogens and Immunity. And I'm here today with Erica Ollmann Saphire, president and chief executive officer of the La Jolla Institute for Immunology, to talk about her work on hemorrhagic fever viruses and SARS-CoV-2. And she'll be speaking about these topics at the Keystone Symposium, Lessons from the Pandemic, Responding to Emerging Zoonotic Viral Diseases. Dr. Saphire is an eminent structural biologist devoted to understanding viral pathogens and pathogenesis. So, Erica, how did your work on lassa virus and other hemorrhagic fever viruses prepare you for the switch to research on SARS-CoV-2 and the current pandemic?
Well, it's very much the programs that we had built and the questions that we had been trying to answer. And the fact that in order to answer the more complicated questions that one lab alone couldn't begin to address, we had galvanized international consortia to put together different labs with different expertise and different samples to answer things at a broader level. And so having had a couple years of experience of thinking through how you might build larger research frameworks to enable the sample sharing and the collaboration that you know you need within a research environment that's inherently built on competition, and how to figure out how to get those things to work. We had kind of a tested and refined and reiterated way of working that allowed us to accelerate a broader, faster study on a novel virus in a pandemic that we needed to. What was good is the willingness of the scientists to put together their samples and their expertise and to build something deeper and more lasting.
Having put together that kind of infrastructure for one set of viral outbreaks was extremely valuable in preparing for the SARS-CoV-2 experience, apparently. With respect to these two different virus families, are there any interesting similarities and or differences that you believe are worth making the audience aware of?
Yeah, absolutely. There's a lot of similarities. So one area is the immune system. We don't know what virus is going to cause the next pandemic. It might be this family or that family. It might be a reemergence of something we already knew. It might be something entirely novel. But we do know the human immune system is what's going to have to respond. And so the body of knowledge that we have built as a field over the last few decades of how do B cells respond, how do T cells respond, what are the differences between neutralizing and non-neutralizing activities in the antibodies, how do you develop a vaccine that will actually give you a longer lasting memory response, the arms of the immune response that you need to put together and coordination of those. All of that will be applicable to the next pandemic too. Even if we dropped down on Mars and found novel Martian viruses, the human immune system would find a way to respond to those. And in terms of the viruses themselves, there is a lot of similarities in how we can approach them. So for a lot of viruses that have a membrane envelope, there's a key, heavily glycosylated, unstable protein on the surface whose job is to attach to your human cell and drive itself in. And because its job in life is to evade your immune system while springing into a much more stable structure to roll downhill in that energetic barrier that allows it to fuse into your cells, you're dealing with a heavily glycosylated, constantly mutating and changing, highly unstable molecule. And that has to be your target for your vaccine design, and that has to be your target for antibodies. And so the body of knowledge built by studying HIV and flu and respiratory syncytial virus and Ebola and so many different pathogens, and figuring out how do you stabilize these kinds of molecules? How do you evaluate the kinds of glycans that are on the surface? Do the most potent antibodies block receptor binding, block cleavage, block conformational change, do something else? How do we assess which parts of these molecules don't change much through mutation? Do some of the more potent antibodies bridge epitopes? How do we develop tools, for example, in structural biology, that let us appreciate the conformational complexity of these things? And what protein engineering strategies do we use to make better immunogens? A lot of the fundamental ideas about how do we figure out how to capture, stabilize, and image the HIV surface envelope could be used and repurposed in trying to understand how do you make an RSV vaccine? How do you stabilize SARS-CoV-2 to make an immunogen that's going to work, or a reagent for structural biology.
It's my view that with SARS-CoV-2, we're extremely fortunate because of the prior outbreaks of other coronaviruses in 2002 and 2003, and then again in 2012, I guess it was, with MERS. We knew, or at least thought we knew, what the likely most important immunogenic target would be, which is the spike protein. But if we had a new pathogen where we did not know what if there was more, especially if there was more than one surface glycoprotein, where we did not know which was the target, do you think it's fair to say it would have taken, or it would take much longer than was the case for SARS-CoV-2 to get a vaccine? I mean, even with mRNA technology, assuming that that was going to be, useful in that particular case.
It would depend on what the pathogen is and how different it is. So for SARS-CoV-2, humanity was really greatly accelerated by the fact that people had been studying mouse hepatitis virus, and had that initial knowledge, or the fact that there was an expansion of study for the original SARS 20 years ago. And the fact that, Jason McClellan had figured out how to stabilize HKU1 and insertion of prolines, and that gave the first ideas to go forward for vaccines, and we can build from there. So it's that foundational knowledge was definitely an acceleration. We wouldn't have mRNA vaccines this year if people hadn't been tinkering with them for 20 years to figure out how to formulate them, how to package them, how to cope with innate immune responses. So things very much build on the shoulders of prior efforts. We have a body of work now looking at adenoviral vaccines or VSV vector vaccines, or what happens if you try to chemically inactivate a virus, or what a subunit vaccine, a protein nanoparticle. And for a lot of pathogens, we don't really know what the right vaccine formulation is going to be. mRNA may work for some things and not others. A VSV vector vaccine may work for some things and not others. So having experience with and approved products of many different types, and some time and relevant clinical experience of what sort of immune response do you get when you use a live attenuated virus versus a protein nanoparticle versus an mRNA, will let us advance multiple platforms next time. And so one thing that was pretty successful, I think, in advancing vaccines so quickly against SARS-CoV-2 was the fact that in parallel, different groups could make and evaluate different things. mRNA, protein nanoparticles, adenovirus, other kinds of vectors, inactivated virus. And if you don't know what's going to work, running lots of horses in the race at the same time is a good way to make sure that some are going to get over that finish line.
So clearly, as a structural biologist, you're focused on taking biophysical information and using that to inform our efforts at development of vaccines and therapeutics. Am I correct that at the current state of knowledge, if we have a neutralizing antibody, solving the structure of the complex of that neutralizing antibody with a pathogen-derived protein, such as the SARS-CoV-2 spike protein, is typically highly informative? But if we just had a crystal structure of such a complex without the biological experimentation, am I correct that we are not at a state of insight where we could predict even the affinity, let alone the neutralization activity?
Well, there's lots of facets there. I mean, you need the biological experimentation to tell you what antibodies you want to bother to take the time and trouble to go and image with X-ray crystallography or cryo-EM. So you know something about them on the way in. You know something about the germline from which they emerged, and then the structure will show you the key residues, the key changes from that germline that tells you something about would a vaccine hope to be able to make that kind of antibody immediately, or is it the kind of thing that might need multiple exposures in a longer period of time? Looking at the structure can tell you a lot of different things. It can tell you what surfaces seem to be key for the most potent activity. Do these antibodies directly block receptor binding? Is that their mechanism? Do they block fusion? Is that their mechanism? Do they bridge subunits together? How important are the glycans? Are the glycans a hindrance to limit antibody access, meaning in a next generation vaccine, you might limit those to improve how the original germline antibodies can bind and start that response going? Or do we need to leave those glycans in because they're key for recognition? A lot of potent antibodies might bridge some glycan to some molecule. So the structure tells you a lot about the roadmap of what the target might be and what modifications you might make in the antigen to better present the kinds of antibodies that look like they're most effective. And if what you want to achieve with your vaccine is broadly protective immunity, not strain-specific immunity, you can look at the kinds of antibodies that are able to recognize multiple related viruses or multiple strains of the same virus and see what kind of places they hit. And maybe you might want to find ways of better displaying those in the vaccine instead of the more mutagenic, but more strain-specific parts. Maybe there are parts of the antigen that you want to block out by adding a glycan. Maybe you want to take key portions that might not be as often recognized by antibodies, but when they do, they give you good response. Maybe you want to just display those in some kind of protein nanoparticle. So that structure is a really good way of understanding what you're making and why and what the end result is. But there's a lot beyond it. So for example, the reason we launched the large Ebola virus consortium is that we had done all the work of figuring out what is the structure of that surface antigen for Ebola and different viruses like it, and where do human antibodies bind that could achieve potent neutralization in vitro. But potent neutralization in cell culture didn't always translate to effective protection in a living thing. And so there are differences. It could be that the kind of antibodies that look fantastic in cell culture just have poor pharmacokinetics in the animal. It could be that their activity is easily recognized in the viro cells that are what you're using in your neutralization assay, but the antibody doesn't block activity in an infection of a different cell type that maybe uses a different receptor or has a different cleavage mechanism. So the antibody will effectively block in that one kind of cell, but in a living thing, the virus has found a back door. And so you need to combine scientists with a lot of different kinds of expertise. You need structure. You need glycan analysis. You need in vivo modeling. You need to understand what you can and can't interpret from the animal models. You need people that can measure PK. You need people that can decide, are these therapeutics even manufacturable? You might have something that you love so much at an academic level, but the protein won't express well enough, or it's just not bioavailable. It's not going to become an effective therapeutic.
Do you see pharmacokinetic differences among antibodies of the same IgG subclass?
Our current consortium for SARS-CoV-2 is evaluating antibodies and antibody-like molecules. And so there are a lot of novel formats of trying to make things multivalent and capture avidity. And some of them are sensational in vitro, but getting the right pharmacokinetics is challenging. Other times there are antibodies for which you might engineer them in some way, or they just happen to have some kind of hydrophobicity or some other stickiness that leads them to cross-react with the target you didn't want to. And it can be quite individual.
Are there any differences between what you found with how antibodies mediate immunity, let's say to lassa virus versus SARS-CoV-2?
A couple of different things. So comparing a large consortium study to, we did one in Ebola, we looked at panels of antibodies against lassa, SARS-CoV-2. And in Ebola, in the fullness of time, we could evaluate lots of antibodies in lots of experiments and lots of criteria. And there's something particular about Ebola that it makes the viral protein that mediates entry is a transmembrane trimer, kind of like you're expecting. But it also makes this soluble, secreted, abundant dimer that can be 80 to 90% of the transcripts of the viral glycoprotein gene. So it's abundantly dumping this dimer. And whether or not that antibody is absorbed by that dimer could be relevant for its therapeutic use. And whether or not the viral particles it's using in neutralization assay had the shape and spacing of that long spaghetti-like filovirus particle, or there was something smaller and rounder, affected the ability of the neutralization assay. The other thing that was key for Ebola virus is that as part of its entry from outside the cell, it goes into the endosome first. In the endosome, it's processed. And that processing removes about half the mass of the glycoprotein. There's one cleavage event that removes these heavily glycosylated domains and exposes the receptor binding core. And so there are lots of antibodies that were all directed against epitopes in these heavily glycosylated domains. These antibodies don't neutralize in culture, meaning they don't mechanically block entry, because what this protein does is it just goes down into the endosome, whether or not the antibody is bound, just by macropinocytosis. And then if after that cleavage event inside the endosome, the antibody epitope is removed, the antibody doesn't neutralize. But those antibodies could do something else. Whereas on the surface of the virus, the surface infected cells, there are epitopes there. And those antibodies bind just fine. By binding, they can recruit Fc effector functions and recruit immune clearance of infected cells and the pathogen. That turned out to be an important facet of protection. And something unique to Ebola virus is the difference between the antibodies that bind this top thing, we're never going to neutralize, but they can recruit Fc effector functions. Other glycoproteins don't drop half their mass in the same way, but it did point to the importance of different complementary roles of antibody in mediating protection in the whole living thing.
That's very valuable to know, I believe. I'd like to move on as we're getting close to the end to some career-related questions. And can you briefly enlighten our audience as to what experiences drew you to a research career and immunology and structural biology in particular?
A research career. You know, I just always thought that science was fascinating. And I didn't think I'd wind up becoming an academic. I remember, you know, in my X-ray crystallography PhD, being 5 years into it and still not having crystals that diffracted. And I decided, I don't know what I'm going to do for the rest of my life. Doesn't make anything at all other than this. But then we found crystals that did diffract. And then the information came in. And then we wrote all these papers. And then like, oh, now I get it. Now I get it. And that's my dissertation right there in purple. I fell in love with structural biology because it was the most intellectually satisfying thing I'd ever seen in my life. And I still remember the day and the seat in the lecture hall I was sitting in as an undergrad at Rice, where the professor explained how you could solve a crystal structure and that you would then be able to understand the XYZ coordinates in space and the V factor of how much it moved from that position of every atom in that protein. And you could see how that information was written and knit into a three-dimensional structure, and you knew how much that structure would move and breathe and change. Anything you would need to know about that molecule, maybe you could figure it out from that. You're just smart enough to figure out how to read it from that. And then what seemed fascinating from there was how to use that structural information to interpret the immune response to pathogens or the immune response to other things like cancer. And that was what originally brought me into immunology, was the incredible complexity of the human immune repertoire and response, and the ability to model and change and create whatever it needed to respond to whatever threat might emerge. And how you might extract from that wealth of possibility tangible molecular meaning about how protection occurred and how you can inspire that in a practical way with a better drug or a better vaccine. That was really the hook.
Excellent. Do you have one or more either scientific or career insights that you believe have been especially important in promoting your research career?
Follow what you believe and what is most interesting to you. You know, people ask, well, what should I have my lab work on? It's something you're burning to do. You know, science is hard enough. There's a lot of long hours. You go into it because there's something that you really want to study and you really want to learn. And that enthusiasm is what's going to get you to try the 1,000th experiment when 999 didn't work. And that enthusiasm is what's going to come out in your words and your grant applications and your talks. That enthusiasm is what's going to attract students and postdocs to your lab. So do what you most want to do. And just because it's not what other people find to be important and interesting right now, as long as you can find a way of conveying what you see, you might have come up with something new. You know, if all scientists do exactly the same thing, we don't get anywhere. You need someone to find a way to ask a new question or make a new tool or look at a new system.
Well, we're about at the end here, so let me just thank you, Dr. Ollmann Saphire, for a highly informative and truly fascinating discussion about how structural biology can, along with other disciplines, inform our understanding of viral pathogens and how they cause infection. Thank you.
Thanks for your time. It's nice to be here.